All right. Hello, everyone. My name is Ian McLaughlin, and I have a PhD in neuroscience from the University of Pennsylvania. And I worked on mapping out some of the neurobiology of addiction and anxiety for my PhD. And so today, I have a very interesting paper to discuss. Maybe more exciting for me than it is for a lot of people, but trust me when I say that it's pretty cool. We're going to be talking about some progress in exploring the use of a compound called Ibogaine for the treatment of psychiatric conditions, specifically addiction. And this will probably be a little bit of a longer discussion than what I've been doing recently, just because I think it's useful to have a sense of kind of like the backstory of this molecule, to have an appreciation of how this group, this research group, got to where they are, as well as an appreciation for how interesting their findings really are. So, Ibogaine. This is a compound that's attained almost mythical status as having unusually effective anti-addictive properties, and not just for one drug. It's been described as being anti-addictive for people who are addicted to everything from methamphetamine to morphine to alcohol to nicotine. And to be clear, this hasn't been sufficiently pursued and characterized in humans for us to immediately deploy it as a critical tool quite yet. But there's no doubt that something interesting is going on with this molecule. And perhaps the most intriguing effects that have been reported in anecdotes of its effects is the alleviation of cravings in people who developed addictions. And, we'll get into the latest developments in just a bit, but if you're here, I presume you want a deeper dive into this molecule, its pharmacology, its history, its effects, so let's get into it. So, to begin, just so we're all on the same page, we're talking about a compound, again, called Ibogaine, which is most commonly derived from a shrub that's native to Western Africa. And in case you're unfamiliar with that part of the world, here's a map to give you a sense of which parts of Africa, or from which parts of Africa it's historically derived. So the plant is called Tabernanthe iboga, which is a shrub, again, that has sort of long, narrow leaves, and it produces lovely white flowers, which then eventually become a sort of bright orange fruit, which you can sort of see in those images. And evidently, the fruit can be either long and tubularish, called fusiform, or it can be round like an orange and called globose. I've never tried any of these fruit, of course, but I can, and I can only find sort of fairly basic descriptions of what they taste like, including like a numbing effect when people eat it, which is kind of interesting to me. But it's not the fruit, it's the root bark of the plant that's most commonly pursued for its ibogaine content, and literally the roots of the plant. So when I'd first encountered this research, Tabernanthe iboga was pretty much the singular plant that was widely known to produce these molecules, at least among the literature that I'd found at the time. And it turns out that there are other plants that produce ibogaine, including Vocanga africana, and I'm probably not going to be pronouncing these words properly, but Vocanga africana, there we go, and Tabernae montana undulata. So both of those plants have been shown to also produce ibogaine. So you might have noticed that I said these molecules rather than this molecule. And so, while ibogaine is without a doubt the most widely studied compound in this family of molecules, which isn't saying all that much, there are other pharmacologically active molecules within the plant that are, worthy of our attention. And so first of all, ibogaine is metabolized into noribogaine, which is basically like an analog of ibogaine upon ingestion. So when you eat it, your body converts it. When you eat ibogaine, your body converts it into noribogaine. But like some other drugs, including the widely consumed diazepam or Valium, the initial drug that's consumed is converted to other psychoactive drugs. So with diazepam, our body's converted to temazepam or Restoril, and oxazepam or Serax. So similarly, with ibogaine, our body's converted into noribogaine. So who cares? Why does that matter? Well, ibogaine and noribogaine have rather different pharmacological action. So noribogaine binds the serotonin transporter with quite a bit greater potency than ibogaine, and it's more active at both mu and kappa opioid receptors, as well as the delta opioid receptor. So that's noribogaine more so than ibogaine. They both have our activity at all of these targets, but noribogaine has that slight difference. So the point is that the molecules to which ibogaine is converted, noribogaine, while it travels through our digestive system, has its own pharmacological activity, which is at least part of the reason that ibogaine ingestion produces incredibly long-lasting effects. It's these potentially excruciatingly long-lasting effects that inspired the latest research, at least in part, that I'm going to discuss in a little bit. And so it's also worth acknowledging that due to the genetic diversity for the genes that encode the enzymes that are responsible for this conversion of ibogaine to noribogaine, it seems that there may be some pretty radical differences in this conversion, right, of ibogaine to noribogaine. So for example, there's approximately 5 to 10% of the Caucasian population that is basically like a complete loss of function in this enzyme that performs this conversion. And of course, that's just one example of one population with this unique genetic relationship to this particular molecule. Others likely exist, and at this point, we're exclusively talking about metabolism. Right? We haven't even gotten into the equally important pharmacogenomic differences between people, right? Where differences among the genes that we all have that encode things like opioid receptors, nicotinic acetylcholine receptors, to which nicotine binds, dopamine receptors, and so on. This genetic diversity can have pretty dramatic consequences for determining the effects of any given drug in any given person. So as a result, there may be some pretty meaningful differences in what this molecule does when ingested by different people. So the more recent history of this molecule is pretty interesting. So the first time this plant was referenced in Western literature was, was in 1864. So the French physician, surgeon, and ethnobotanist Griffon du Bellay, I believe, might be how you pronounce that, described this plant as an aphrodisiac and a stimulant of the nervous system. So Griffon du Bellay, by the way, that's his last name, and I won't even commit the atrocity of trying to pronounce his first name. You can see it down here, though. And so it was named Tabernacle iboga in 1889 upon the first botanical description of the plant. And then an extract from the root bark of Tabernacle iboga, again, those plants, was designated as ibogaine in 1901. So evidently, ibogaine was pretty widely available in the 1930s, particularly in the form of a tablet called Lambarine in France. So it had about 8 milligrams of ibogaine in it, which is a pretty low dose compared to the doses that are taken, more recently, by some people for its anti-addictive properties. And it sort of like mind-blowingly became popular as a performance enhancer among athletes, which is kind of cool. It's a story I'd love to explore a little bit further. I couldn't really find very much explanation as to why, but anyways, evidently it was also sold in France as a treatment for psychiatric conditions, but it was eventually taken off the shelves due to adverse effects. So, beyond the purported performance-enhancing effects of the compound, there were claims of its anti-addictive properties that stretch back to the 1980s. So studies began to show that ibogaine reduced morphine and cocaine intake in rats, and alcohol intake was reduced in mice. And it also seemed to mitigate the manifestation of morphine withdrawal symptoms. So, pretty interesting pharmacological pedigree there. So there are a few reasons that this molecule is so near and dear to me. So first of all, it was the topic of the first research paper I ever wrote when I decided to major in neuroscience in college. It gave me an opportunity to dive into a whole body of literature that most college students and even PhD candidates never really explore. Like most biomedical scientists probably don't even know that this stuff exists. Even at that time, in the early 2000s, the pharmacology of the molecule was so complicated that I was clearly out of my depth. And it seemed like every neurotransmitter under the sun was affected by this molecule. So second, once I started grad school, because I decided to join a lab that focused on the neurobiology addiction as one of its core focuses, I ended up chatting with people who had family members and loved ones who were struggling with addictions to various substances. A few times people would independently bring up ibogaine, asking if they ought to travel to Costa Rica or Canada in pursuit, of a treatment with this molecule because it was so illegal in the United States, about as illegal as it could be. So for me, this suggested two things. First, the discussion of ibogaine had grown to an extent that people who, never explore these topics in neuropharmacology, they would encounter it on the internet. So, it clearly wasn't so niche anymore. Okay. And then, so the third reason that I have a soft spot for this molecule, I attended a talk by Rick Doblin, the executive director of an organization called the Multidisciplinary Association of Psychedelic Studies, or MAPS, in Boulder, Colorado. This was many years ago. It must have been around 2007, 2008. And while the talk he gave was focused on the organization's ambitions to support the investigation of potential clinical applications of psilocybin and MDMA, that was the focus, for the treatment of psychiatric conditions, Ibogaine was mentioned. And by the way, some progress has been made in exploring the potential clinical utility of both, psilocybin and MDMA, including a clinical trial that's just about to begin, exploring whether a combination of psilocybin and Suboxone might help to treat addiction. And so I have some videos on both of those substances, which I'll link in the description below. And then so finally, as I always try and say when I introduce myself, I focused on mapping out the neurobiology of addiction and anxiety in graduate school. It was part of the neurobiology. And by that, I was trying to identify how specific neurons in a specific part of the brain contribute to both anxiety and addiction, and particularly the anxiety that may predispose some, to addiction, as well as the anxiety that people experience while, withdrawing from drugs to which they become addicted, right? So I had written my little college essay before I'd known that I'd end up in a lab with a long track record of exploring addiction. And after finding my sea legs in the lab, I wrote a couple of review articles and book chapters on the topic of addiction, mostly nicotine, but not exclusively, and specifically the role that this circuit that I was studying plays within addiction. So, while writing one of these publications, years after that college essay, I happened to come across a body of literature that explored whether a molecular relative of Ibogaine, of the Ibogaine molecule, so a different molecule, but a congener called 18-methoxycoronardine, or 18-MC. This literature was exploring whether or not 18-MC might also have some anti-addictive properties. And essentially all of this work was done in the lab of a scientist named Stanley Glick in New York State. And so, frankly, when it comes to, that's Stanley, Dr. Glick, when it comes to, the potential clinical application of Ibogaine and its molecular relatives, in the treatment of addiction, particularly for, the basic neurobiology of addiction, Stanley Glick is easily one of the most important scientists to the sustained interest in this molecule. So had he not done his work, I suspect that we wouldn't have seen this paper that I'm about to discuss. And so Glick's lab had produced some of the earliest preclinical evidence that this congener of Ibogaine, 18-MC, seemed to produce anti-addictive effects in preclinical models, in animal models. But what was particularly interesting about this lab's work was that it seemed to produce these results for multiple addictive drugs, right? So like we had been hearing about when it comes to Ibogaine, anti-addictive properties for morphine, alcohol, methamphetamine, nicotine. It seemed like that might be the case here too with 18-MC. And interestingly, it looks like there is a clinical trial exploring the potential application of 18-MC for the treatment of addiction. And it looks like they might be getting ready to begin recruiting. So, you might want to check it out if you're interested. That's not the topic for today, but I hadn't known of this clinical trial until I read into the main, I did some reading, for the main study for today. And I'm not going to get into, my own perspectives on Ibogaine and 18-MC specifically, but it's pretty interesting. And so I figured I'd include it. And best of luck, right? 18-MC is in a phase 2 clinical trial, which suggests they've already demonstrated enough safety to progress to the next stage, safety in humans to progress to the next stage. And this phase 2 is devoted to figuring out if the treatment is actually effective as a target treatment. So that's pretty interesting and definitely something I'll be keeping my eyes on. Also, if you're interested, there's a bit of a documentary that has Dr. Stanley Glick describe his interactions with a man named Howard Lotsof. And so I'll put a link in the description below because it's pretty interesting. It's kind of old, but it's pretty interesting. Another part of the interesting history of Ibogaine. On the topic of documentaries, if you're interested, Vice did a short documentary on Ibogaine a few years back that you can check out, and they focused on a specific form of treatment for addiction with Ibogaine that involves a bit of ritual and sort of culture and stuff, which I assume is not the method of treatment that this group who conducted this study that I'll be talking about in a second really have in mind. But anyways, I also have a link to the Vice documentary in the description below. Anyways, so you might be wondering why so much effort has gone into finding analogs or congeners of Ibogaine. Well, apart from it being fairly difficult to obtain, the effects of the compound can last from anywhere from 15 to 36 hours or longer. And honestly, I have to assume that this wild range of effects may be attributable, at least in part, to the fact that there's no like standardized exposure to Ibogaine. You can go different places and you'll be treated in different ways. So, some people take a small dose early on, like a test dose. Then they take a subsequent dose or doses over time. And so, if you were to space out those doses, that would inevitably produce, very different effects relative to someone who just took a large dose all at once. And, I have to assume that the folks in France who were consuming lambarine or lambarine weren't experiencing the types of effects for which Ibogaine is sort of notorious. And so what are these notorious effects? Well, never having been exposed to the compound myself, of course, I can't personally explain it to you, but fundamentally, it induces significant hallucinations and dissociation. And I've seen it compared to compounds in the psychedelic family of hallucinogens. So, while the more widely familiar members of this family of compounds, drugs like LSD and psilocybin, last summer between, 4 and 10, maybe a little bit longer with LSD, 4 and 10 hours, we're talking about something that can last almost a day, if not several days. So you go to sleep and you wake up and you're still under the influence of this substance. So, both this like rather extended duration of effects, but probably mostly the hallucinogenic effects of Ibogaine, as well as a few other issues with this molecule that we'll discuss in a second, have inspired groups to explore whether, rather than using Ibogaine itself, similar molecules based on the Ibogaine molecule might be developed that could deliver, similar effects. Well, okay, let's get to it. So a study was published a few days ago that explores exactly what I think a lot of people in neuropharmacology were hoping would be explored when it comes to Ibogaine. So, inspired by Ibogaine, they worked to figure out what the molecular characteristics of Ibogaine, like, the specific arrangement of it, the atoms in its molecule, which specific components are central to its purported anti-addictive properties. And so it's a method called, function-oriented synthesis. And you can think of it this way. So, we're all familiar with keys to unlock doors, right? And there's a specific pattern of teeth height and length and ridges along the pointy end of the key or the blade of the key, ridges, notches, and teeth. And a specific combination and sequence of these characteristics is designed to specifically and theoretically exclusively open a specific lock. So those characteristics are like the features of a key that are like minimally necessary to open a lock. Without those features, you can't open the lock, and you basically, you have a different key. So the part of the key that you hold in your hand, though, the head, evidently as it's called, is largely unimportant to the key's ability to open its lock. You can shape the head like a rectangle, like this one, or you can have a shape like a circle or an oval or like a guitar or something, whatever you want. When it comes to, the specific purpose and goal of that key, the shape of the head makes no significant difference to the ability of the keys, the ability of the key to open a lock. So neuropharmacology functions somewhat similarly. So, specific molecules are able to interact with specific receptors throughout the brain, and that's because the molecules have specific shapes, specific atoms that are able to interact with different areas of a receptor that would normally be binding, neurotransmitters like serotonin or dopamine, GABA, glutamate, and so on. But there's one key difference. So when it comes to the abilities of various molecules to interact with receptors, one molecule can interact with multiple receptors. So it's sort of like having a bunch of different locks throughout your house, right? Each room having a unique lock and a unique key to which it corresponds, but there would be some keys that could open a handful of the locks and not all of the locks, while another key could open a different set of locks and not the other ones, right? So, this gets a little complicated, because if we were to like really compare the pharmacological targets in the brain to locks, it would be more like having like a lock that has multiple entry points for our keys. And each of those entries does like a slightly different thing when each key is inserted and twisted. But, this is pretty much where the lock and key heuristic of pharmacology, pharmacological action starts to kind of break down. But anyways, all this to say, when it comes to molecules that bind specific receptors, just like how only a specific part of the key is necessary to unlock a door, only a specific part of a molecule is necessary to interact with a receptor. You don't need all of it. The other stuff can modify the ability of that molecule to get to the receptor, to get into the brain and stuff. But if you don't have that one key component of the molecule, you can't interact with that receptor. So, if you're interested in figuring out which part of molecules are fundamentally necessary, that blade of a key that's necessary to interact with the specific receptors or pharmacological targets in the brain, it can be pretty hard work to figure out that perfect combination of molecular ridges, notches, and teeth, right, that are needed in a molecule to achieve an intended specific effect. So given the evidence that ibogaine has some anti-addictive properties, perhaps we could figure out which part of the ibogaine molecule, again, may be responsible for those anti-addictive properties. And therefore, perhaps we could make a molecule that only delivers those anti-addictive effects, or mostly delivers those effects, and not the hallucinogenic effects. So that was the main goal of this group, who published in the publication Nature, which, in case you're unfamiliar, is one of the most prestigious possible, publications for scientific research. It's extremely difficult to get your work published in Nature as a scientist. And if you go check out the paper, of course, I put the link in the description, you might also notice that there's a ton of names that are listed as authors in that title. So, that this seems to be increasingly common for these types of studies that get published in outlets like Nature or Science. But this, many names, tends to indicate that there is a ton of collaboration between different labs. And that's not always the case when there's a ton of study authors listed, but that does seem to be the case here. So the scientists who contributed to this series of experiments were all over the place: universities of California in Davis and Santa Cruz and in San Francisco, but also the University of Colorado in Denver, along with several private institutions. So in other words, there was a lot of teamwork that went into this publication. So I mentioned that ibogaine is fairly well known to have these hallucinogenic effects at certain doses. However, there have also been reports of fatalities associated with ibogaine over the years as well. And there's speculation that it might be attributable to cardiac arrhythmias. And I'm unaware of any more specific details regarding like a mechanism for this potential hazard of ibogaine. And it seems to only occur in certain people. But these events have been recorded. And in 1993, the U.S. FDA, the Food and Drug Administration, or FDA, actually approved a clinical trial to study the effects of ibogaine itself. Believe it or not. But a female study participant unfortunately suddenly passed away during these tests. And these types of investigations were basically abandoned in 1995 by the U.S. Biomedical institutions, and the compound basically languished in Schedule 1 hallucinogen limbo, right, for years and years. And so as a result, most of the human data that we have is anecdotal and derived from like those gray market addiction treatment centers in countries outside of the United States, like Costa Rica and Mexico, and I think Canada. And so, therefore, we really don't have particularly like high quality data that characterize if, when, and why the compound may be hazardous to some people and not others. So, I thought it might be worth briefly discussing some of the evidence that ibogaine itself may present some toxicity in some cases, and this evidence is a little bit problematic. So at high doses, like 100 milligrams per kilogram, rats showed general signs of potential neurotoxicity. However, at lower doses, around 40 milligrams per kilogram, it didn't end up with inducing the same effects. So those are both wildly high doses, in these neurotoxicity studies compared to what I've seen reported humans taking. So, to give you a sense of a ballpark dose of ibogaine that's taken by humans that people tend to ingest, and of course it's not like ibogaine treatment is particularly standardized, right? There's going to be a lot of variability here, but dose ranges I've seen are an initial test dose, right, exposure something like 2 milligrams per kilogram, and then that's followed by something like between 10 and 20 milligrams per kilogram, so substantially lower than the toxicity doses that were evaluated in those studies. And that said, frankly, I'm very skeptical that these doses are rigorously standardized and quantified. Like, I'm skeptical that they know that somebody's taking only 10 or 20 milligrams per kilogram. Just based off of the journalism I've seen covering ibogaine treatment centers, I just find it hard to believe that the actual doses are accurately identified, that they even have the capability to do that. But anyways, so I'm going to exercise a little bit of discipline here because I've already been going on for a while. But and I'm not going to go into all the various studies that have been done over the years that explore the potential toxicity of ibogaine, particularly since we're going to be focused on this novel compound, or actually series of compounds. But to suffice it to say that there's evidence that ibogaine isn't necessarily completely benign, isn't as benign as I think a lot of people perceive it as being. And it has some pretty unique pharmacology that really puts the molecule in a class of its own when it comes to these mind-altering substances. And so it was both the evidence that ibogaine seems to have this potentially anti-addictive activity, as well as the evidence that it might be dangerous at certain doses for certain people, that inspired this group to explore this, an alternative molecule that may be able to deliver the same benefits without the hazards. So, while reading this paper, I was introduced to a word I'd not seen before, psychoplastogen. Psychoplastogen. So the concept is that certain molecules are able to induce a state of elevated neuroplasticity for a limited period of time. Specifically, these potential medications would make it so that neurons ultimately form new neurites, which are basically like finger-like projections. Let me just make sure that, yeah. Okay, yeah, whatever. They're sort of like finger-like projections that extend from neurons, the things that make neurons look like neurons. And those projections enable neurons to communicate with one another, theoretically resulting in the creation of new synapses throughout various parts of the brain. And so to give you an idea of what this looks like, here's a video of some neurons in a petri dish. And it's animated, and you can sort of get a sense of how neurons kind of explore their surroundings with, the potential of forming new synaptic connections, new points of communication between different other neurons. And so, to be clear, this is a process that's guided by biochemicals that are released from other neurons. So you can, see those sort of circular central bodies from which all those very long fingers extend. That's the cell body, or the soma. And you can see that these long fingers seem to be like frantically exploring the environment around them, right? Well, there are receptors, similar to like neurotransmitter receptors, on the ends of those fingers. And they're basically looking for molecules that other neurons are releasing as a sort of like candle in the window, right? Sort of like it's a signal saying, hey, I'm here, let's chat, right? Let's form a connection. So when they say psychoplastogen, they're suggesting that these substances, and most importantly to their work, something that they called tabernanthalog, which is kind of adorable, increases the rate at which neurons exhibit plasticity. And secondly, it's that effect that may be central to why we've been, increasingly finding that these substances, these mind-altering substances, are useful to help people who are looking for new addiction treatments, new alternatives. And, the group did some fun experiments to explore just how much, their series of Ibogaine analogs induce that kind of plasticity. And you can see it down here. These are, just neurons that are sitting in a petri dish, bathed in, a hospitable and cozy bath of nutrients and ions and stuff, the things that neurons need, and are made to glow this sort of lovely electric blue color. So that's not like what they naturally look like. They're sort of treated so that they glow that way. And then, within the bath, a petri dish bath, a series of various drugs are introduced to compare how effective they all are at, basically convincing neurons to send out projections to each other, or doing it more often. So ketamine, for example, is thought to be a psychoplastogen. And it's recently been approved, by the way, by the US FDA, the Food and Drug Administration, as a treatment for depression, as well as an analog of ketamine, S-ketamine. So, I don't think it's been conclusively demonstrated that this psychoplastogenic effect, this increasing of neuroplasticity effect of ketamine is critical or even relevant to its antidepressant effects that it exerts. Though it's definitely one of the leading theories as to like why it's helpful for some people. But it's not the only sort of like, whoops. Oh, okay. Awkward. It's not the sort of like only molecule that's been associated with both antidepressant effects as well as acting to promote neurite growth. It's not the only ketamine. It's not the only mind-altering substance that's been identified as a potential psychoplastogen. But then you can also see ibogaine, or IBO, as well as a few other ibogaine analogs down on that side. And they're compared to both ketamine and just the control, over here. It's a solution that it's like a neuron solution that doesn't have any, interesting molecules in it. And you can just sort of like qualitatively tell, right? The neurons that are treated with basically like all of these molecules tended to send out way more projections in some cases, and way more in specific cases, like with ibogaine and ketamine, right? If you compare that to the control neuron right here. So what this group is proposing is that, this ability of some molecules to promote this kind of structural change in neuronal circuitry, specifically the circuitry associated with addiction, right, and drug-seeking behavior, that ability of these molecules may be like the central benefit of these compounds. So rather than like the intense, subjective hallucinogenic effects that they induce, it's the increased neuroplasticity, particularly in certain parts of the brain that they hypothesize, they enumerate, that's delivering this therapeutic effect, not the intense like hallucinogenic subjective effects. So to be clear, this is a Nature paper, and so this wasn't the only figure that they included to explore this effect of these compounds. But I felt like it was the most intuitive. If you just look at it, you don't even really need to know very much about neuroscience. You can just tell this one's a lot pokier than that one, right? And it's also cool looking, right? Not the least of which because this was the kind of figure that I used to make in the lab. So anyways, you might remember our friend Stanley Glick. Nope. There we go. Well, one of his central hypotheses to explain the potential anti-addictive properties of Ibogaine was that it might just block the interactions that habit-forming drugs like morphine, methamphetamine, or nicotine have with various receptors. It might just like get in the way or prevent those drugs from inducing their action, and so they're just not as fun. They're not as enjoyable. This group, however, was focused on that psychoplastigenic effect of Ibogaine. And what they set out to do was engineer an analog of Ibogaine, a molecular cousin, which they call their Ibogalogs, which is kind of cute. They were trying to make these molecules that don't have the potentially toxic and hallucinogenic effects of Ibogaine, but do have its anti-addictive properties. So in other words, by tweaking the molecule here and there, you might get to the desired effects and avoid the undesirable or unnecessary effects. So of course, this paper gets into the medicinal chemistry to explain like which parts of the molecule they honed in on, which they determined to be interesting and important. But, imagine that you're looking at it, one way to think of this strategy is like, imagine you're looking at a table that's like lined with legs all along the perimeter. It's just like a straight up wall of legs, right, on all sides of this table. It stands, but, it's effective as a table, but if you're interested in figuring out exactly which table legs are absolutely necessary, and only the table legs that are absolutely necessary to keep the table standing, you might just start removing a leg, one at a time and seeing what happens. Does it fall? Still standing? Remove another one. Evaluate, modify, repeat until you get exactly the effect that you're pursuing without destroying the table's ability to stand. So, and then you might even then start exploring ways to modify those table legs to make an even better table, more durable, reliable, stable, and so on. How to make a better table by modifying those table legs. So this is a clumsy metaphor, I admit, but it's it's a basic idea of what they did. And ultimately they landed on a specific molecule, which they call tabernanthalog, Ibogaine Analog. Got it? That seems to be safer and less toxic than Ibogaine itself, and they have some evidence that it's not hallucinogenic, but that's not the strongest evidence. So here's where I think we ought to be at least a little bit reserved regarding the non-hallucinogenic effects that they claim that the molecule has. So they're relying on the behavioral responses of there we go. Zebrafish. So nothing against zebrafish. They're beautiful little animals, but the evolution of the human nervous system departed from our common ancestor with zebrafish, quite a long time ago, something like several hundred million years ago. And so, that's not to say we can't learn anything from zebrafish. Quite the contrary. Like I've, cited zebrafish studies and behavioral studies in my own publications. But just that when it comes to an effect like hallucination, I wouldn't rely solely on zebrafish behavioral responses. And of course, the authors, in the study aren't implying as much even. They just want to be totally clear about it. They also included some studies involving mice, their head twitch response specifically. But they did some really excellent studies with zebrafish using techniques that were used in the lab in which I worked to assess the potential for these molecules to induce seizures. So it's worth, it's not worth getting into this technique, which in short was basically calcium imaging. It's kind of complicated, but they found that neither Ibogaine nor tabernanthalog, their molecule, induced seizure-like activity at the doses that they were exploring. So probably one of the group's most important findings were that tabernanthalog, their analog of Ibogaine, seems to have the potential to reduce both alcohol and heroin-seeking behavior, which, as I said earlier, is one of the most intriguing characteristics of Ibogaine. It seems to have anti-addictive properties for completely different habit-forming substances that share little pharmacological activity. I feel like I'm getting a little dim here. Right? Let's do. Sorry about that. So it doesn't adjust very well on the fly. Sorry about that. So, pretty much all habit-forming substances that are used recreationally share at least some central pharmacological activity, like the mesolimbic dopaminergic circuit. And I've gone on for too long, so that'll be a topic for another day. But anyways, finally, what does this mean for you? So, first of all, there really aren't particularly effective treatments for addiction. There are some resources available, of course, and they're meaningfully effective for some people, but outside of psychosocial treatments. However, like social support networks for people who are trying to stay abstinent, biological or pharmacological resources are pretty limited. And while there are some interesting efforts to explore whether some treatments that are used to treat like opioid addiction might actually be helpful for addiction to other substances like alcohol, the majority of medications that are used to help people who are trying to kick a substance habit are only used for specific substances, like disulfiram or antabuse, acamprosate or Camprol, or perhaps ondansetron. I always mispronounce that one. Zofran. Those are used for alcohol addiction. Bupropion or Zyban or Wellbutrin, varenicline or Chantix, or just straight up nicotine replacement products like nicotine gum or nicotine patches for people who are trying to kick a smoking habit or nicotine habit. They're used for specifically nicotine. And then, methadone or Suboxone for opioid addiction. Of course, these are all medications that are used specifically to treat addiction to specific habit-forming drugs. And so, there's some interesting efforts to see if again, some of the medications for opioid addiction may be useful for alcohol addiction. But the reality is that all of these treatments aren't nearly as effective as we all hope they would be. And, while we're all focused on the imminent threat to our lives, right, and of the lives of the people we love, COVID-19, the reality is that there was an epidemic in the United States before COVID-19 came around. So the last time I talked about these stats were where I talked about how we're failing to confront addiction, the reality of addiction in this country. The CDC hadn't yet compiled the data from April of 2019 to April of 2020. Now they have, and tragically, unsurprisingly, the stats have gotten even worse. And, we'd already seen higher rates of overdose fatalities between March of 2019 and March of 2020 than we'd ever seen in the history of the country. But April of 2019 to April of 2020 surpassed that record. And so we're in dire need of new solutions to this public health crisis. And this group is doing exactly the kind of work I've been hoping might happen. Not only did they find some preclinical evidence that an analog of Ibogaine might be both safer and as effective as an anti-addictive treatment, anti-addiction treatment, but it's even quite easy to synthesize from Ibogaine. It's easier to transport. It's not as hydrophobic as Ibogaine, so it could be particularly useful. And then beyond that, they provided some evidence that supports the hypothesis that the anti-addictive properties of this whole family of unorthodox treatments for psychiatric conditions in general, hallucinogenic molecules, and particularly addiction, may be attributable to their effect of producing a state of increased neuroplasticity in some key parts of the brain that may help people who are trying to kick habits that are otherwise nearly impossible to kick. So it's exciting. And I'll definitely be keeping my eyes peeled for any future progress that this group makes. Perhaps moving to more complicated preclinical models, more complicated animals. But we shall see. But it's exciting. I can probably take this down. Anyways, yeah, that's what I have for you today. So I know this was kind of long. So if you're still with me, I'd appreciate if you might subscribe if you're not. But also I'd appreciate any input that you might have as to how I can make these better. There were a lot of things that I didn't go into, like the story of Howard Lotsoff that I mentioned earlier, who was perhaps the leading proponent of Ibogaine for quite a while in the 70s and the 80s after having kicked a heroin habit along with several of his friends, following an Ibogaine experience. He even patented Ibogaine as a treatment for heroin and cocaine addiction, smart move, and ended up treating 30 addicts from all over the world to some success. And he was also a major factor in motivating NIDA, the National Institutes of Drug Abuse here in the United States, to support investigations into Ibogaine before they were abandoned. And there are even some videos of the guy you can check out on YouTube. Anyways, I should stop. All right, why don't we start the live half? Wait, let me make sure that this doesn't look like garbage. Boop. What if I do this? Boop. That's better. I'll do that. All right. Hello, everybody. My name is Ian McLaughlin, and I have a PhD in neuroscience from the University of Pennsylvania. And just now on YouTube, I talked about a new study that just came out a couple days ago that explores, first of all, why it might be, that explores a molecule called Ibogaine. So if you're familiar with sort of some of the more unorthodox treatments for addiction, you might have come across Ibogaine. It's a very interesting molecule. It has some downsides to it, not the least of which being it induces about 15 to 36 or more hours of hallucinations. And it's potentially toxic in some people at certain doses. But the innovation, the reason this is an important study, is that they seem to have developed an analog of Ibogaine, a sort of molecular cousin of Ibogaine, that might present some of the anti-addictive properties without presenting the potentially dangerous properties. So pretty cool. So check it out. Anthropoid, youtube.com/anthropoid. Hello, Irene. Okay. Question: did you hear about the new analog of Ibogaine that does not induce hallucinations? Yeah. So Javier, I just talked about that on YouTube, so you can check it out. It's kind of a longer one, I'm sorry to say, because I've been interested in Ibogaine now for years. So yeah, check it out. Do you know my wife Candace? Dallas Cowboys 408. Do I know your wife Candace? No. I don't think I know anybody named Candace, actually, weirdly enough. All right. What are your thoughts on VP shunts? Do you need them all your life? That's honestly a question for honestly a question for a physician. I don't know. Are you offering to see if it can fit in my mouth? Oh, thank you, Daryl. Okay, question. Let me do this. It's so dumb that I have to constantly do that. Nope. Brilliant. Good job. Question. Can cannabis induce the kind of dreamlike state that Ibogaine usually does? Can cannabis? No. Well, I mean, so cannabis definitely probably has. Hey, Amanda. Cannabis, I think, has more significantly mind-altering properties than I think it's sort of widely appreciated as having. Hello from Philadelphia. I used to live in Puerto Rico. But when it comes to like the hallucinogenic effects of a lot of molecules, not just Ibogaine, they have a unique set of pharmacological action that cannabis does not have. So, you know, briefly the pharmacology of cannabis is that they bind cannabinoid receptors and endogenous cannabinoid receptors that would typically bind neurotransmitters that we make in our bodies, and endocannabinoids. Whereas, you know, whereas the effects of like a classic psychedelic drug like psilocybin or LSD principally bind serotonergic targets. So these are like pretty separate. I mean, you know, they overlap because cannabinoid receptors regulate a lot of neurotransmitter receptor systems, but that direct effect on serotonergic activity is probably, I mean, it's very uncontroversial to suggest that that's probably the reason that they have those potently mind-altering psychedelic effects. So the pharmacology of Ibogaine is very, very complicated, and it's actually not that well understood, interestingly. And in fact, one of the scientists that I respect the most recently said that on Twitter. He's like, we still really don't know how cannabis works, or how Ibogaine works. But this group, central to what this group discovered, well, okay, so first, but Ibogaine has, you know, we know it has this very complicated pharmacology. It interacts with like almost every neurotransmitter system you can name, including serotonin. In fact, one of the early theories as to why it was that Ibogaine has these anti-addictive properties was that it might normalize the serotonin receptor in the intracellular component of the serotonin receptor. And the nicotinic acetylcholine receptor, which was, you know, an interesting thought. You know, it would be unusual, but you know, it's theoretically possible. But anyways, yeah. So just because of that distinct pharmacology, despite the fact that cannabis, that cannabis, you know, has these sort of pseudo psychedelic effects. I suspect that the sort of hallucinogenic state that Ibogaine would induce, I've never been exposed to it, so I can't tell you for sure, but just from what I've read and what I've seen, that it would be pretty distinct. You know, it'd be like comparing, you know, cannabis to you know, full-blown dose of LSD or something. So now, the therapeutic effect of Ibogaine is what is part of why this study was pretty cool, and probably part of why it ended up in Nature, which is one of the best publications that you can publish science in. And it was that it seems to induce a state of heightened neuroplasticity. They call it a psychoplastogen. And they demonstrate, and I included at least part of their figures on that, effect on neuroplasticity. But part of why so that would be an interesting finding if it was just about Ibogaine and just about the analogs that they synthesize. However, they also compared it to ketamine. So ketamine is another hallucinogenic drug, not a psychedelic. It's oftentimes called a dissociative anesthetic. Totally different pharmacology from Ibogaine, from psychedelics, but widely now appreciated to be pretty effective for the treatment of depression in some people, particularly severe depression. And they show that both ketamine, and it's also known to have this psychoplastogenic effect. It elevates neuroplasticity. But so they show that, hey, ketamine has this beneficial effect. Ibogaine is thought to, there's a lot of data to suggest that it has this beneficial effect on anti-addictive effect. And so maybe it's this. Maybe it's like, it doesn't really matter what specific neurotransmitter systems, these molecules are targeting. Rather, it's that it just, by different mechanisms, induces a state of heightened neuroplasticity. And so therefore, particularly if somebody's also receiving psychotherapeutic treatment, right, for depression or for addiction, that might just render one's brain more sort of ripe to being modified by treatment. So yeah, okay. Is there a possibility of there being neurotransmitters in the human brain that we have not discovered? Oh, sure, yeah, absolutely. Not only that, I mean, there are debates as to whether or not certain neurotransmitter receptors exist. Like the sigma receptor is this sort of like kind of mysterious receptor that for a long time was essentially theoretical. And in fact, it was one of the receptors with which, staying on the theme of mind-altering substances, with which DMT interacts. Still not thoroughly understood, and there are definitely people who argue that there's no such thing. It's very, very difficult to prove that, particularly now, to prove that like a not particularly numerous, receptor exists in the brain. If you don't know what to look for, how are you going to find it, right? You don't even know what questions to ask. But yeah, that's always an ongoing, effort, particularly these more complicated neurotransmitters, or let's just call them signaling molecules, because it's sort of a pointless distinction. The more complicated ones, peptides, maybe ones that are released by glia that can influence gene expression, and then also like mechanisms of interneuronal signaling. Like I think I talked about it last time. Like neurons can just release packages of genetic material, mRNA, just release it, and then it's taken up by other neurons. So yeah, definitely. Okay, are viruses man-made? Viruses can be man-made, and does COVID-19 affect the brain? Great topics, love it. So first, viruses can be man-made. I made many viruses, and it's actually easier than you might think. At least for certain kinds. Certain kinds of viruses are easier to make than you might think. Some are actually rather difficult. Like it's actually kind of laborious to make a rabies virus or a herpes simplex virus, but they're very useful viruses because of how they evolved, because they're able to essentially move along our nervous system in very specific ways. So, like how do you get injured by rabies? Apart from being bitten by something that has rabies, the virus actually infects your, sensory nervous system and essentially crawls up the projections that extend from your spinal cord, crawls up those projections until ultimately gets into your spinal cord, and then crawls up into your brain and then begins wreaking havoc. And that's why if you get bitten by a rabid animal and the bite is farther away on your body, it'll take you longer to develop, dangerous symptoms than if you're bitten like right on your neck. Best to avoid rabies if you can. I wouldn't recommend catching rabies if you can avoid it. Now, so does COVID-19 affect the brain? So it's a really interesting. I think you would have to define making a virus. I don't see us doing it from scratch. Yeah, okay. Sure, that's actually a really good point. So first of all, making viruses is I say it's easy. It's because you're not like crafting the virus. So a virus is just a very, very simple sequence of genetic code, right? You can have DNA, ones that are made out of DNA, ones that are made out of RNA, and they come in all different shapes and sizes. COVID-19, for example, is relatively large, actually, for a coronavirus, but it's relatively small relative to other genetic entities, right? I think it has something like 30,000 base pairs or something like that, compared to our like 3 billion, right? So it's rather small. But, all a genome is just a set of, instructions. And those instructions essentially command various, mechanisms within cells to occur, or to stop occurring, right? Synthesize more protein, synthesize less protein, these kinds of things, right? Generate new synaptic connections. So when it comes to making a virus, essentially what you do is you have a, you essentially co-opt bacteria. So you basically turn bacteria into factories of viruses. You get certain genetic material, and then you infect bacteria, and then those bacteria will be able to package up viruses, and then you extract the viruses that then explode out of the bacteria by like spinning down the stuff, the soup in which the bacteria were growing. Okay, so that's basically the process. Okay, so now, does COVID-19 affect the brain? So it's a hot topic right now. I mean, there's a lot of evidence to suggest that yes, it does. The question remains, however, how? So we don't know if COVID-19 actually directly gains entry, or SARS-CoV-2 is the virus that causes COVID-19, actually gains entry to the brain. That's not an easy thing to do, even for viruses. Most viruses can't. But viruses don't have to do that to indirectly affect the brain in deleterious ways. And we think that's probably, for my money, I think that's more likely what's happening with the neurological symptoms that have been observed for COVID-19 patients. And so maybe I should describe this a little bit. So fairly early on, so meningitis isn't a virus. So meningitis is just describing a state. It's describing the inflammation of your meninges. Different viruses, different bacteria, different things can cause meningitis. But yeah, no, I mean, for sure, parasites can gain entry to the brain. I'm not saying that they can't. It's just that there are more viruses that can't than can. Okay, so evidence that COVID-19 affects the brain. Fairly early on, once COVID-19 had spread outside of China into Italy and into France and into basically Southern Europe, there began to be reports of people coming into the hospital or their clinics presenting what looked like neurological symptoms, and only neurological symptoms. So confusion, stroke, these types of things. No fever, no cough, none of the sort of things that we were initially instructed to keep an eye out for. And in fact, to such a great extent that there were hospitals in Italy that set up dedicated wards to COVID-19 neurological COVID-19 cases, to care specifically for these neurological patients. And then eventually there were reports like that in the United States. And there were reports of people who would come into the hospital, again, with confusion, akin to something like dementia, and then, but not present any of the coughing, any of the fever, any of the classic, symptoms that, again, we were keeping an eye out for. And so it became common enough that even the CDC has included sudden confusion as a symptom to be vigilant of occurring, right? That if there's sudden confusion, that might be an indication that COVID-19, that you're looking at a COVID-19 patient, and not just like a stroke patient or something like that. But it can be very, very difficult to distinguish. Okay, so, but evidence that that SARS-CoV-2 can gain entry to the brain. So before COVID-19, before all of our lives changed fundamentally in unpleasant ways, there was SARS, right? SARS-CoV-1, right? The sort of first major coronavirus outbreak that happened in the early 2000s. Stayed mostly in China, but it did spread a little bit. And some studies were done on patients who had passed away from SARS, and it turned out that they were able to identify SARS-CoV-1, the SARS virus particles in the nervous system tissue of those patients. So it's unknowable as to what mechanism that was, but that's some evidence that it could potentially actually be able to gain entry into the nervous system. And while I said most viruses can't gain entry to the nervous system, the ones that can, there are a bunch of different strategies. So, I mean, you might, a virus that could just bind any of a variety of receptors that are along essentially the blood-brain barrier could theoretically be internalized into the brain. And so that's not impossible to conceive, particularly given what we think is the evolution, the mechanism of evolution that has rendered SARS-CoV-2 so much more effective than SARS-CoV-1, which is that it's able to interact with it seems, a wider variety of parts of the body, of receptors in the body, which would enable it to infect a wider variety of tissue, a wider variety of parts of the body. And that's why you get things like, multi-organ failure, weird blood clotting tendencies, vascular pathologies, heart attacks, strokes, that perhaps explains why SARS-CoV-2 is so much more promiscuous. Okay. So any thoughts, any long-term study of Adderall and Lexapro? I'm 47 and take both. I'm not sure that there's a specific study of the combination of Adderall and Lexapro specifically. There are definitely long-term studies of Adderall and SSRIs and Lexapro. Boop, 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 boop. Right. So yeah. Lexapro and SSRI. But I don't know about the combination. So on first pass, like if a family member were coming to me and asking like, hey, should I be worried? Wow, that's quite a sunset, huh? Should I be worried about this combination? So long as the doses are not excessive. I mean, there's no reason that anybody would ever take excessive SSRI doses. Like there's no recreational benefit to it, right? Or appeal. Adderall, however, is, abused by some people, or at least people take higher doses because it does have, recreational appeal. It is amphetamine after all. But the pharmacological actions of Adderall and an SSRI like Lexapro are pretty distinct. You're tapping into pretty different monoaminergic neurotransmitters. You're tapping into serotonin with SSRIs obviously, and dopamine, catecholamines, dopamine and norepinephrine with Adderall. And so just so long as there is no overdose, I wouldn't be concerned about the combination. But first of all, if you're concerned about long-term consequences of taking both of these substances, you should definitely bring that up with your physician. And so, long-term studies have been done on both, and they seem relatively benign over the long term. Again, presuming that they're being taken at reasonable doses. Hey, from Finland. Hello, from Philadelphia. If COVID-19 was handled with real leadership and wasn't politicized, it would have never gotten this bad. I think that there's a lot to that. I don't think you're wrong about that. And I think that there's a pretty obvious tendency in the country to, put all the blame of this, what is objectively horrible mismanagement of this pandemic, on just one person, on the president. And, of course, the buck stops with the president, right? It is ultimately the president's responsibility to manage all of this, but there were just systematic failures all along the way. And I really do hope that there's going to be and there will be, an autopsy once we're on the other side of this thing, that, identifies, just to give you one very specific example. So I remember when the guidance was coming out of the CDC and the Surgeon General's office to not wear masks, or to not go out and buy masks. And had they only said, we're worried about having insufficient supplies for, frontline, clinical workers, that's fine. I mean, that's just objectively true. It happened. But they said that they weren't effective, that they don't actually do anything. And for me, that was like a big red flag. And I remember having conversations with my family, with my parents, because of course that's reported. And who am I to contradict the CDC or, the Surgeon General's office? Come on, they have the best and brightest, some of the best and brightest scientists in the world, working there. Not necessarily at management, but career people at those institutions. I told you I made viruses. I wore a mask making viruses. It's just standard. And it just from a kind of a scientist understanding of how viruses are transmitted, and particularly respiratory viruses, it was like how could you possibly justify saying that these aren't effective? There weren't any studies. So that was a red flag, and I don't know who deserves the blame for that, but somebody deserves some blame for that because that was just absurd. And that wasn't the only failure, right? Okay. Any truth behind large prefrontal cortex and intelligence correlation, or is it a myth? So Cedric, yeah, I mean, it's an interesting topic for sure. When you say like a large prefrontal cortex, there's certainly a correlation if you're thinking evolutionarily, right? If you're thinking like, if you're comparing like why are humans, why are Homo sapiens more intelligent than bonobos or chimpanzees, our closest genetic relatives? Our brains are actually rather similar. Our brains are bigger. That's one thing. But for sure, one of the primary anatomical differences is a field called comparative anatomy. One of the most overt differences is our giant swollen frontal lobes. That includes the prefrontal cortices, or the prefrontal cortex. And so if you're thinking evolutionarily, yes, for sure, there's a correlation between, larger prefrontal cortices and greater intelligence. However, if you're thinking like just within humans, this person is intelligent, that means that they must have a larger, or they're more intelligent than I am, so they must have a larger prefrontal cortex than I have. That's not quite how the brain works. So just having a large single part of the brain, generally speaking, I mean, it may be helpful, but generally speaking, that's not going to be associated with like preternaturally, great intelligence. Rather, it's probably going to be associated with some kind of pathology. Some kind of anomaly that makes it so that other parts of the brain aren't functioning properly. So just to give you an example, like let's just imagine that a wild scientist, or I, developed a way to expand the size of my skull just a little bit, and then expanded the numbers of neurons in my prefrontal cortex. Well, what would likely happen with the prefrontal cortex? One of the main things it does is that it sends inhibitory signals to more primitive, more evolutionarily ancient parts of the brain that regulate things like emotion, sexual urges, libido, appetite, these types of, they're very sort of foundational urges that sort of guide our daily behavior, right? The prefrontal cortex is able to send, the prefrontal cortex receives a bunch of signals from the hippocampus, from the neocortex, from all over the place, integrates all of that information, and then sends inhibitory signals to those mood regulating parts of the brain. And so that's very handy, right? Because of course, most of our urges are probably not prudent, right? You probably, every time you walk by, an ice cream store, pizza store, or whatever, pizza shop, pizza restaurant, I guess, if you didn't have that prefrontal cortical inhibition, that those signals from the prefrontal cortex, then you'd probably just buy a pizza every time and then get an ice cream on top of it and all that kind of stuff, right? Because you wouldn't have that inhibitory signaling. However, if you just had overly abundant prefrontal cortical inhibition, that might correlate to just increased anxiety or increased behavioral inhibition, where like you just fail to engage various opportunities or various, opportunities that emerge in the moment, right? Because you just have excessive inhibition of those primitive drives that are, while they can be problematic, they're also critical, right? They're critical to reproduction, for example. They're critical to, these types of things, right? So, across different species, yes. Across humans, not so much. Now, having an unusually small prefrontal cortex, that's different. So having there's no such thing as like an average sized brain, there's no such thing as like an average human brain. Like, you could never find a person that has the, perfect middle of like the bell curve of like variability in neuroanatomy among humans. There's no such thing, right? That's like a theoretical concept, but imagine that exists. Somebody who has an unusually a smaller than average prefrontal cortex, that might be associated with, that might manifest as like intelligence deficits, or overly impulsive behavior. I think you're legally required to study a guy named Phineas Gage if you take a neuroscience class or even a psychology class, who basically lost. It's a funny story, but he lost a huge part of his left frontal lobe including his prefrontal cortex, and he became this very impulsive guy, very aggressive and stuff like that, whereas prior to his injury he wasn't those things. That's like the classic story, but there are other examples where just, disrupting the prefrontal cortex correlates with behavioral issues. So yeah. You'd be a great teacher. I would love to be a teacher, but I need to support, help support my family, and I don't have that kind of patience. Interesting. Thank you very much. Oh, that's my pleasure. Thanks for asking. I love that kind of question, the like neuroanatomy question. Why can I not stop masturbating? Is it a disability? So Chris, chronic masturbation is not a disability so much. I don't know if it'd be qualified as that. Yeah, Phineas Gage. Good job. Good spelling. I'm surprised you knew how to spell it. That's cool. Yeah, there's great videos online. He's been studied for a long. There's actually some problems with how his story has been exaggerated a little bit, but this central principle is sustained. Another one that you might be interested in, just on the topic of the relationship between neuroanatomy and human behavior, human intelligence, is Charles Whitman, who was a mass murderer. He was the guy who climbed to the top of the university, the clock tower at the University of Texas in Austin, and just began randomly shooting people with a rifle. And it turned out that he had this very large tumor in a certain part of his brain. So I think you would also find that fascinating. Part of intelligence is considering the end product. What is humanity's end product? Yeah, Daryl. Well, as I always, try and argue that human intelligence, it's really awesome. I'm so glad that we have it, but it is an evolutionary experiment. We shall see if it was evolutionarily advantageous to develop this big fancy intelligence with the possibility of us not being intelligent enough to prevent ourselves from hurting ourselves, right? Sidelows, whom is larger becomes more intelligence. I'm not sure what you mean. Oh, but are we though? I see what you're saying. Yeah. Is it true it's easier to learn as a child versus as an adult? And do you stop growing your brain after 25? So first of all, yes. I mean, so the first one, yes, it is easier to learn a variety of different things as a child than it is as an adult. For example, I have a daughter that's almost 2 years old, almost her birthday. She was born on December 31st. Pretty cool. So, I have this young daughter, and she learns words like almost immediately now. Like, I'll say, what is this? We're looking at a picture of a fire truck, and it has a hose on it. And she'll be like, what's that? That's how she asks. And I'll point at it and say, hose. She'll just remember that now, forever. It's just so wild. Hey, signatures. I think it's been a while. Good to see you, man. And here's the reason. So her brain is almost like the early era of the Earth, where like the crust was being completely changed constantly. There's tons of volcanic activity. There's magma everywhere. It was just constantly changing. Sort of like how her brain is right now, meaning it's filled with magma. No, meaning that neurons. First of all, there's way more excitatory activity. Meaning like neurons are just more excited. That's part of the reason, by the way, for why. Is it your birthday? No, so that's my daughter's birthday. She was born 6 days before I was born. Big gap in time though. But yeah, I'm on January 5th. She's on December 31st. Anyway, that's part of the reason that elevated excitatory activity in the brains of children. Part of the reason for why some people can grow out of epilepsy. Because over time, that excitatory tone, the level of excitatory signaling decreases. So that's part of it. But the main reason is that tons of synaptic connections between neurons are being formed and gotten rid of, and it's just this constant sort of cascade, this ballet of neurons forming new connections with one another that didn't exist before, or they're strengthening connections more rapidly. It's just a far more dynamic organ than adult brains. Now, adult brains are different. Like adult brains are still dynamic, but they're just not as dynamic. Like right now, there are new synaptic connections that are being formed. I don't know how old you are, but yeah. So there's that. Then what was the other part? Oh yeah. And does your brain stop growing right after 25? 25 is about when. 25, 26 is about when that very rapid sort of pubertal or pubescent era of rapid changes in the brain begin to kind of reduce. Yeah, so 23. Yeah, so you're still pretty young. So I mean, you're still able to learn, let's say, more rapidly than I am. I am 32, almost 33. Oh my god. Anyways, so there's that. So even as you get older, like, the brain is still dynamic. You're still forming new synaptic connections, and it's just more slowly. So that's why, there's sort of like this ripe period for a child to learn a new language when they're young, and it just becomes so much older. And in fact, one of the interesting manifestations of this, by the way, is people's accents. So, like when somebody moves from, let's say, Russia to the United States, one of my colleagues, one of the scientists whom I respected the most, moved to the United States, actually moved to Italy first and then the United States when he was in his late 20s. He had that classic Russian accent, that, charming Russian accent. And but because he moved at that age, basically what happens is that at a certain age, your ability to particularly consonants, your ability to generate sounds, like particularly those that are in English that have a bunch of weird ones, like the R sound is very strange, becomes so much more difficult as you get older and older, to the point where, there's a certain point where, like, some languages will basically just like co-opt one of their own sounds as a substitute for, a sound for a different language. So, when an accent is sort of solidified, that's an example. It's like a demonstration of that, like, reduced dynamism within the brain, synaptic dynamism. Look at Albert Einstein's brain. It has huge side lobes. So, U.S. performance. So that's not quite the anatomical distinction in Albert Einstein's brain, though you're correct that there are some anatomists that argue that there was something very unique about Albert Einstein's brain. It's worth noting, however, that not everybody agrees on that. So there are some people who argue that that's actually not true. And part of the problem is that Albert Einstein's brain was stolen after he died by some dude. It was like literally taken from him, and then it was lost. Just languished somewhere, I think under a desk in a jar. It was like not preserved as well as I would like to have had it preserved. Right, and then they found it. But it was preserved in this like really sort of haphazard, well, not haphazard, but just not optimized way. And so as a result, the tissue isn't like, I've seen slices of Albert Einstein's brain, and it's like, it's preserved worse than like this brain slices I preserved were, right? There's some room for, improvement there. But so as a result, it's kind of difficult to like get very, very precise about his anatomy. But the argument isn't that he had like oddly large temporal lobes, which is what I assume you're referring to. Rather, it's that he has actually a truncated sylvian fissure. So basically, it's like a sulcus is like one of those, you look at a human brain and it's wrinkly, right? You have peaks and valleys. A sulcus is a valley. And one of the arguments is that he has a truncated sulcus. That sulcus that divides the temporal lobe from the parietal lobe is truncated, meaning it's not, it's like shallow. There are some other ones, like for example, some people argue that he had like unusually low aggregates that were detectable in his brain. But from my perspective, it's like, is there going to be something unique about Albert Einstein's brain? Yes, there definitely will be, but there's something unique about my brain, about your brain. No two brains are going to be identical. Even identical twins will have very, very minute, but existent differences between their molecular neuroanatomy, if you want to call it that. But, so every human will have a unique brain. So for me, it's like, I don't know that we need to appeal to like a specific anatomical, particularly gross anatomical, like an anatomical difference that you could see with your eyes, explanation for why he was brilliant. He was brilliant. Some people are brilliant, and through chance and circumstance, he received the kind of education. He grew up in a certain culture. He grew up and then moved to other cultures that enabled him to flourish. Blah blah blah. All these things conspired or collaborated to make for a very, very brilliant and unique person. And so it may be true that there were these unique anatomical features of Albert Einstein's brain, but not necessarily those aren't necessarily the things that would explain his genius, because he was brilliant. Period. Right? Yeah. I'm 47. It's over for my brain. I remember watching you at 29 years. It's been a while. I know it has been a while. Yeah. Yeah, well, I finished my PhD. So that happened. Synaptic connection become loose. That makes learning abilities become more hard. That's what I know. So when you say synaptic connections become loose, so actually, like, I guess I'm not 100% sure what you mean by that, but like the thing that makes, the thing that makes, not the thing, one of the things that makes learning more difficult as we get older isn't that like synaptic connections deteriorate. In fact, probably it's the opposite. It's probably that, synaptic connections are too cemented, are too static. And so, that you're just less likely to be able to adapt to new information. Yeah, thanks, Signature. Yeah, I don't know if this was a while ago, but I appreciate that. I learned Japanese from 18. Just to give you another example. So, it's not like learning isn't exclusively about like the formation of new synapses, although it's important. It's also about like neurons themselves changing. So neuroplasticity is operative at multiple levels. It's not just formation or removal of synaptic connections. There's also the insertion of new receptors into pre-existing synaptic connections. There's a removal of some. There's a change of the receptors themselves. Are they more sensitive, less sensitive? There's like the formation of, like, you can imagine synapses, a synapse here. This neuron, if this synapse is particularly active for whatever reason, and this is a certain kind of neuron, it can form another synaptic connection on that synapse. So it's like communicating at two different points. It can form a synaptic connection on the axon that is hosting this synapse. So there's multiple levels of neuroplasticity at play here. And one example of one thing that gets worse as we get older is that ability for receptors to be moved around a neuron. You have receptors on your neuronal cell body or soma. You have neurons along your axon. You have neurons. You have receptors along your, the somata, the soma, the neural cell bodies. You have receptors along the axons. You have receptors on synapses and on dendrites, of course. And those those move. Those are mobile. And so as we get older, just because literally the composition of the membranes of neurons changes, receptors are less able to migrate and move, and that can be a huge problem. Well, not a huge problem. I mean, that is natural, first of all. It's actually regulated by cholesterol, believe it or not. But that just makes it so that your brain just doesn't change as rapidly, doesn't adapt as rapidly. Doesn't make it impossible to learn new things, of course. It's just that's the difference between why young people, cholesterol, certain forms of cholesterol. That's why children are just more effective at learning. Herd myelin is how learning sets once set. Oh, I see. How like learning is cemented, right? How can or cannot myelin be undone as habits reversed? So when you say like myelin is how learning is like, let's say, established, I suppose I could understand what whomever conveyed that to you meant. Probably what they mean is that when synaptic connections, let's say, when a new axon is formed and a neuron is communicating with a new neuron that it didn't before, that is one mechanism by which learning can occur. And that axon probably would be myelinated. So myelin is basically just a cell type. It's a fat, right? It's basically goo that makes it so that the signals from a neuron are far more capable of moving, of literally traveling from one neuron to another through a mechanism called saltatory conduction, the Latin for saltar, to dance, because the signal sort of bounces along these little myelin. So if you can imagine in your mind, imagine like your phone cord or whatever. It's like wrapped in that plasticky stuff that insulates the electricity inside, right? So if you could imagine the way myelin works is that if you could imagine just little breaks along the cord, so it's not completely contiguous, it's like these little sets that wrap around the axon. And so the signal, like when a neuron fires an action potential, the signal literally bounces. I say literally, I mean sort of literally bounces along those little gaps called the nodes of Ranvier. Anyways, so like if you have a new axonal growth, it will be myelinated, and that is a way to cement. That's one way to think of it, that learning being cemented. But learning is a byproduct of a variety of different levels of neuroplasticity. In fact, the video the thing I did on YouTube just before this, youtube.com/anthropoid, you can You can find it on my Periscope profile. I talked about how Ibogaine, a theory for why Ibogaine and ketamine, by the way, may be effective in treating depression and addiction. Really beautiful study. And basically, one of the things that these molecules do that are essentially unrelated. I mean, they're both hallucinogens, but ketamine is completely different from Ibogaine. And then they made a new molecule that's based off of Ibogaine. That's like the point of the paper. But anyways, they basically demonstrated that these totally unrelated molecules induce a state of heightened neuroplasticity, which may therefore render various treatments more effective. So you can check that out, youtube.com/anthropoid. I have friends who have treated their addiction with Ibogaine. That's very interesting. If you're on Twitter, I would really appreciate it if you would reach out, and because I would like to ask some questions about that. I found Ibogaine interesting for a very long time. Basically it was the first paper I ever wrote in college was on Ibogaine, when I decided to take the plunge into to become a sorcerer, a neuroscientist. Check that out, youtube.com/anthropoid. May I come on and ask questions? Of course. Oh, if you mean call in, absolutely, please. What year did the Great Depression start? Lay off the caffeine. What? You lay off the caffeine. Reading and writing with pencil makes your brain produce more cells. This is an everyday duty. I think I know the studies to which you're referring, that like the actual motor behavior of we're getting a call. Motor behavior of writing may make it easier for you to learn things. We can talk about that after this. So, is your name Archie? And if so, where are you calling from?
Madison, Wisconsin.
Oh, cool, from Wisconsin. What's on your mind?
So I was actually asking a very interesting question that I've always wondered. So in school, everybody's seen them. Everyone knows that they're everywhere. And, it could be like something that happened in the womb or whatever. I don't know what really causes disabilities. But have you ever wondered why or how is it possible that people with severe disabilities, Stephen Hawking, I suppose he had like a generational kind of thing where he was just born with, was that ALS or something?
Yeah.
Yeah, so that's a little bit different, but have you noticed that there's people who have like, what's it called? They have cerebral palsy, or they have it where they have Down syndrome or something like that, and they just seem like there's certain things that they obviously struggle at, but there's also things that they're very, 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 very good at. So can you explain that? Like, how does that work?
Sure. Yeah. I mean, so that's also true for some cases of autism, where people have just Asperger's. Yeah, right. So Asperger's is sort of on the autism spectrum disorder. And so first of all, I think you're right to sort of put Stephen Hawking in a different category, just because first of all, he lived most of his life, or not most of his life, but a lot of his life without the disability. And then, of course, and he received the best education probably in the world at the time, during that period, and then was essentially confined to living a life of his mind, right? So I think that's slightly distinct. But what you're referring to is, the conditions to which you're referring. Let's focus on autism. They arise due specifically to unique brain architecture, unique neuroanatomy, right? So, for example, with, autism, there are the sort of classic social deficits, for example, right? Those arise because certain parts of the brain, I don't know that they've been thoroughly mapped out, are distinct, are just, built differently. And so, while we tend to, when, you're meeting somebody new, if you're looking at me right now, your eyes, believe it or not, probably are focused on my eyes. Maybe you're going down to my mouth. You sort of bounce between those major things, right? For somebody with autism, they don't have that they don't exhibit that bias. You can track what somebody's looking at. And so, they'll just sort of drift, particularly children, they'll just sort of drift around the face. So, that capability is different. I mean, it's sort of registered as a deficit because, it's how humans interact with one another, but it's not the byproduct of damage or anything like that. However, so you have that social deficit, but that unique brain architecture is also going to manifest in other parts of the brain that have to do with computation or memory. And so as a result, my favorite theory to explain why autism arises.
I think I'm gonna change my bio. I forgot to do that a long time ago. It's probably super dirty, so I apologize, but please don't hate me.
No worries. Don't worry about it, man.
I think it comes off Twitter, and Twitter is definitely not for the faint of heart. At least not on my page.
Agreed. Well, I definitely had many years of misspent youth myself, so no hate here. But anyway, so one of my favorite theories to explain autism is that early on, as our brain is developing, in the first few years, our brain, again, is very, very dynamic. We're making new synapses, getting rid of old ones, and that process is regulated genetically. And so if you inherit a certain form of a gene, that process is going to occur in a slightly different way. And so different sets of synapses will be pruned, is what it's called, will be pruned than would be if you had a, let's say, more typical, form of that gene. And so as a result, you're going to have a variety of differences, one of which being, in, let's say, the temporal junction, right, where we know a lot of computation occurs. And so as a result, these folks, while they have social deficits, may also have tremendous gifts in, computational processing, like mathematics and stuff like that. And so it's just the same mechanism manifesting in two different ways.
So it's just easier for them to focus on one certain thing. Do you think that that's what makes it, well, then some of them, it's actually impossible for them to focus. So some of them, they're just very, very, very good at focusing, like, if you say language arts, such as like, learning Microsoft Word, Microsoft Office, writing books, things like that. They can spend a lot of time and stay focused on something pretty easily, but only just like, am I making any sense there? What do you think?
Yeah. I mean, essentially, like functionally, I think you're basically right. Like, you can think about it this way. I don't know if you play video games. I just started playing this game, Cyberpunk. It's pretty awesome.
Oh my God, I love that game. I love that game. It's so crazy how you can have the girl and then have it have a dude's voice. That's what mine has. It's a girl character with a dude's voice. And then it even has a penis. I'll just say, like, I think
that's
it's
wild. Yeah, I know.
But,
that's very funny.
Yeah, and I wish that they'd let you actually see your character without you actually having to go into camera mode, every other game, but they want to stick to a first person. But it's a really interesting game.
Yeah. I'm thinking I'm going to, like, Twitch stream it or something. But anyways, you're building a character, right? And you have different, numbers that you can assign to different categories, right? Attributes. And so, if you think of somebody who is what we call neurotypical, somebody who doesn't have any other, any conditions, that it's just the same number all the way down, right? It's just evenly distributed. For somebody, who, inherited these sets of genes that rendered their brain to be unique in various ways, it's sort of like cranking up the numbers on some of these attributes and then, having deficits in others. So it's sort of like the same mechanism is responsible for that unique brain architecture that then manifests as, social deficits, anxiety, these types of things, sometimes compulsive behavior, obsessive compulsive disorder, but then also tremendous gifts, not always, but sometimes tremendous gifts in certain categories. So that's the basic mechanism.
Okay, and another quick thing, what do you think about people who have extreme anxiety? You can't just assume somebody with anxiety has autism, can you?
Oh, no, yeah. So I actually wrote my PhD on the neurobiology of anxiety. So anxiety is extraordinarily complicated. It's not as simple as, maybe how we think of being fear, because anxiety involves learning and memory, involves past life experiences, it involves, and then of course mood, a lot of the same circuitry as fear. So for somebody who has severe anxiety, it's true that anxiety is a component of a lot of psychiatric conditions because mood is involved in so many things. It's involved in every single decision you make, obviously every emotional experience you have, but every decision you make, every thought you have is in some way influenced by mood. And so as a result, there are certain neurons within the brain that are responsible for generating anxiety, for heightening anxiety. Anxiety is an evolutionary mechanism, right? Without anxiety, we wouldn't be as likely to engage in various behaviors that, are unpleasant in the short term but pay off in the long term, or avoid dangerous, situations. But those same neurons that enable us to, sit down and study for, a test for like a week because we know we're anxious that if we don't, we're going to get a bad grade, and that'll be bad because bad things will happen to us. Our parents will get mad, or we won't be able to have the life that we want, blah, blah, blah. So for, those neurons are involved in a variety of things. So that's why you get, some people with autism spectrum disorder are highly anxious. Some people with depression are also highly anxious. But then also, some people with schizophrenia exhibit anxiety, paranoid anxiety. So yeah, it's the activities of specific sets of neurons. It's a network of neurons, not just one part of the brain, where for one reason or another, past life experience, genetic inheritance, that the baseline activity of those neurons is just cranked up a little bit. And so people are just predisposed to anxiety.
Would it be possible to ever take medicine to get over it? I mean, to take medicine for the short term and then get over it yourself, like mentally, because isn't it a mental thing versus physical?
Well, they're one and the same. I remember people would always make that distinction that, certain addictions are chemical or physical, whereas some are just psychological. Everything that is psychological is also physical, right?
Yeah, that's true, because it can control. Yeah.
Well, because the brain is producing all of it, right? And so now your question, could we ever make a treatment selectively targeting anxiety? That was my main goal. And, it's obviously harder than one lowly PhD could accomplish. But yes, I think theoretically, once we map out those specific neurons to which I'm referring, what they are, then we can find out which receptors that normally bind neurotransmitters are unique to those neurons. And then we make a molecule that binds to those receptors.
Yeah, because I used to take what was it called? It was
benzodiazepine.
Damn it. What was it?
A benzodiazepine, like alprazolam or Xanax or Klonopin.
It was actually clonazepam. So they didn't give me the good stuff. So they gave me the sister drug of Xanax and stuff. And I maybe shouldn't have asked for Xanax, because that's probably why they gave me the weak stuff. But because I just said, I heard Xanax really works with anxiety and stuff. So, and I asked for it, and then, they should have really given it to me. But at the same time, they didn't want me to get addicted. So maybe I was displaying traits that would have made it seem like I would abuse that, which I really don't. But, that's probably what they were thinking. But
yeah.
Either way it goes, I just really wanted to not have anxiety anymore.
Yeah.
Imagine being in a room with a lot of different people, and everybody is still approaching, you seem somewhat pretty approachable, but you still feel alone in an entire room full of people.
Yep.
You know what I mean? So it's you may not know that, but there's a lot of people who suffer through that, and that's what anxiety is. I'm consistently thinking that, I'm doing something wrong, or that I'm doing too much, or I'm not doing enough, or, should I be the first person who texts after hooking up with somebody, or should I wait for them to text me? There's just a lot of things that happen, and it's too much to think about. I don't know. I
could not agree with you more. Honestly, it is this thing, again, that evolved to keep us alive, right? And to motivate us to engage in behaviors that are beneficial for us and the people we love, our kin and ken, right? But yeah, I think just because our brains basically haven't changed for tens of thousands of years at least, if not longer, if not hundreds of thousands of years, this ancient organ that was sculpted by evolution in the savannas of Africa is the same organ that's being used to have this conversation right now and to navigate social situations like in high school and stuff. And so you're going to get these moments where, the neurons that were active in your brain that were cranking up that anxiety might have kept you alive in the savannas of Africa. But actually now they're deleterious. Now they're a problem. And so yeah, so Klonopin, it used to be Klonopin, clonazepam. The main difference between Klonopin and alprazolam, which is Xanax, or diazepam, which is Valium, is that it's just a shorter acting, right? But they also have rapid sublingual tablets, which are so it gets into your bloodstream a little bit faster. But anyways. They used to be very commonly prescribed for anxiety, but the problem is that they're very habit forming, and right. And the withdrawal from benzodiazepines is actually one of the few drugs that people use recreationally that's actually potentially fatal. The other one being alcohol.
Is there a withdrawal from clonazepam as well?
Yeah. Only if you get to a certain dose, like level number of milligrams that you're taking regularly. If I think a standard dose for Klonopin is something like 2 milligrams, I think. Somewhere like that. And so if you're taking 10, 15 milligrams or something like that, you have a huge tolerance to it, in other words, then if you immediately stop, you run out or whatever, you can have seizures and potentially, and that could damage the brain, but then potentially also be fatal. Weirdly, one of the only legal recreational drugs, federally legal, also does that, and it's alcohol.
Wow.
Yeah, isn't that wild?
Well, I mean, and that seems like the effect, like they say that the effect is the same thing as alcohol, like, with clonazepam and things like that, but I don't think it ever felt that way to me, in my opinion. Now, there's this other thing that's like this anti-anxiety medicine. It's like I don't think it's over the counter, but it's like, I forget what it's called. It's a very light type of thing, and basically it makes your brain, the best way to explain it, like it pops, it wakes you. I don't know how to explain it, but it's like it's a anti-anxiety medicine that basically is something that doctors feel a lot more comfortable prescribing instead of just straight up Xanax and stuff like that. So I forget what it's called, but I took so much. I haven't taken medicine for over 2 years now, but I took all that stuff. There's also this, I'm so sorry, but what's it called? Like this depression medicine?
Like Prozac, fluoxetine, or Lexapro.
Lexapro, that's what I took. I took Lexapro. And the problem with that is it really messed with libido.
Yeah,
You could not, and it's like, imagine being very, very horny, because, I mean, that's really, like, you can even read my bio. Like,
we're primates.
It is what it is. But either way it goes, it's like, imagine being very horny and not being able to do anything about it.
Yeah.
It's like, it was so bad, like, it's like, and then I would literally be sitting there, excuse my French for this, but I'll just say attempting to relieve myself for hours, because you just can't get anything out when you're on that stuff. So I'm like, it's not even worth it. I'm just going to sit here with this depression and anxiety and deal with it instead of taking the medicine, because that's how bad it was.
Yeah, man.
Like,
Yeah, no, absolutely. I mean, it sort of sounds silly because we like rarely talk about these things, openly with people we just met, but like that is a huge problem. Like it's a very, very significant issue with those medications, because we're primates. We evolved to enjoy that part of our biology, right? And it's sort of central to our lives. So yeah, that is one of the motivations where we have to develop better treatments for depression and anxiety. But at least we have those medications. But there's room for improvement. But anyways, listen, Archie, thanks so much for calling in, man. A lot of really interesting topics. I hope to talk to you again.
Thank you so much, man. I appreciate it.
Take it easy. Oh, and enjoy Cyberpunk. I'm trying to set up Twitch, but it's so unintuitive. It's terrible. Are you telling me I should drink? I'm not telling you should or shouldn't. Being loved from someone, also very helpful. Yeah, so US Performance, I mean, that's right. And one of the ways that we express that love is through intimacy. And, it is true that these medications really do disrupt that biology. Opioids do that too, by the way. So Daryl in Edmonton, I believe, right? What's on your mind?
Yes, hi. Yes. Yeah, actually, 2 points I wanted to discuss with you. The first point was, I was thinking about this myself, because as I understand it, when a person forms an addiction, it molds the very physical architecture of the brain. So, and I understand that like most drugs really more or less have like an effect on whether neurotransmitters are absorbed or reabsorbed by their neurons. So my question is, to reverse addiction, wouldn't you have to ultimately undo that physical architecture of the brain?
Yes. I mean, yeah, at a very fundamental level, yes. But I mean, you could say the same for treating depression or treating anxiety or treating any psychiatric illness, is that, you will need to, the goal of therapy, the goal of treatment is to, well, the goal is to reduce suffering, obviously. But, fundamentally, the way that happens is through some kind of physiological changes that occur in the brain. When it comes to addiction, and this is what I talked about on YouTube just a bit before, youtube.com/anthropoid, one of the interesting theories that's becoming more popular is that some of these novel treatments for addiction and depression induce this state of neuroplasticity, elevated neuroplasticity in certain parts of the brain. And because of that, other forms of treatments become more effective. They sort of take hold a little bit more readily because of that elevated neuroplasticity. That let's say neuroanatomy that had been sculpted by years and years of substance use is able to be more rapidly modified by treatment or just by abstinence. And so yeah, I mean, 100%, you're right about that.
And my second question, and I'm not sure if it's really a question that can be answered, because it's to do with like myself personally, and you're not aware of the details of my diagnosis, but I mean, there's a lot of evidence to support that I'm a high functioning autistic, but I also suffer from like generalized anxiety and chronic depression and attention deficit. So it seems to me that it's somewhat uncanny that all these pathologies should manifest randomly. Like, it seems to me that there must be like a frame of architecture that's giving result to these different pathologies, that they're all interconnected and producing these various pathologies. What do you think about that?
Yeah, so basically, like, there's some shared physiological substrate for all of these symptoms that you experience. Yeah, I mean, well, sure. I think that's true. I mean, at a fundamental level, there must be, right? That if somebody exhibits high functioning symptoms of a high functioning autism, whatever changes occur, and we have an idea of some of the genes that are likely involved, likely also implicate more than just the sort of classic symptoms of autism, the social deficits, these types of things. These systems are all regulated by, at least at some point, a shared regulator, and so as a result, you get changes in all those brain regions that otherwise aren't necessarily as directly connected. Of course, they influence one another, but they're not like a direct circuit. But they're all influenced by a shared genetic, upstream regulator.
There must be like I know this would be like a maybe not the fitting analogy, but there must be like a primal operating system that's flawed on some level, and then it's spreads out into like these different pathologies.
Yeah, man.
Like a tree and how it branches off is what I meant to say. Like, yeah.
They share a branch.
Yeah. Because I would find it like it just doesn't make sense to me that like there would be like something irregular or abnormal about different parts of my brain. That there has to be some main frame that's contributing to these different pathologies. So you would agree with that consensus?
Yeah, pretty much. I mean, I wouldn't necessarily frame it as, I suppose this probably sounds like a little hokey to some folks, but I sincerely do think of it this way, that like it's not like a diseased or like a fundamentally damaged mechanism that causes these unique patterns, neuroanatomy, right? It's just a different one. And, there are some gifts that are conveyed with this, and there are some challenges that are conveyed with this. It's
my understanding that now, within science, there's this evolving debate that we're just neural atypical, that we're not necessarily damaged, for lack of a better word, because people with high functioning autism, me being one of them, will often say, like, I really do not feel like there's something wrong with me.
Right. Yeah, totally. I mean, particularly, there are cases of autism spectrum disorder that are debilitating, right? That it's essentially impossible to function, right? So that's not what we're talking about here. Yeah, absolutely. And so the question for me isn't like, let's correct whatever unique mechanism, unique event, biological event occurred to, result in high functioning autism. Rather, let's figure out ways that enable people with these, these atypical, this atypical neuroanatomy, to exploit their benefits, their unique benefits, and enable, but still have a high quality of life, by helping to reduce the severity of whatever deficits they experience. But like fundamentally, like, so I'll give you a more sort of tangible example. So, synapses, as our brain is growing, from the womb all the way up to right now, or developing, let's say, the formation of synapses relies on, specific sets of proteins that interact with one another, so that this synapse sort of, locks in almost. They don't lock, but they interact, and it's like, oh, okay, we're here. A little bit like Velcro, kind of. And so those are proteins, right? And those are encoded by genes. And so there are unique forms of those genes. And so as a result, like, it doesn't matter where in the brain synapses are, there are synapses there. And so if you just if you just modify the genetic sequence for those little Velcro proteins, you're going to have different kinds of synapses. In some cases preferable, in some cases, less preferable. But like your fundamental concept that, like, these symptoms of ASD likely branch from a shared fundamental biological mechanism, like, branches spreading off of a central branch. That's correct. I mean, that's a good way to think about it.
Yeah, because it's my understanding that, like, genes don't necessarily, like, you don't necessarily have, like, specific, like, fingernail genes, for example. I mean, those genes might be tied to other activities that naturally occur in the body as a result of those genes. There are
very few parts of our biology that are the byproduct of a single gene. There are some, for sure there are some.
Yeah.
And when I say parts of our biology, I mean, like, that we can see, right? I mean, obviously, like, every protein has its gene, right? But the expression of every gene is influenced by other genes and by other proteins, and by parts of our genome that aren't even expressed. And so, a bunch of factors sort of all come together to influence everything from height to, fingernail texture. I think that might be one of the weird, monogenic ones, but it's all like the byproduct of a constellation of genes interacting.
And I guess what I mean is that the expression of a single gene doesn't necessarily just have consequence for, like, one specific part of your body. It could have consequences for different parts of your body or different parts of its development.
Yeah.
So it's like tweak one thing, fine tune it, and then something else becomes unwounded and untweaked.
100%. That is one of the concerns about CRISPR. That's widely discussed. It's not a secret or anything. Where we have an idea of which genes are involved in ADHD or various things. I mean, there are some companies that claim that they can give you the genes that encode for intelligence, and so. But let's just take ADHD. Generally speaking, imagine you're a parent and you go to a physician or a genetic counselor, and imagine living in a world where they could say, it's Gattaca. Basically, it's like living in a world where you could say, I'll take that form, I'll modify this gene so that they have no predisposition to addiction, to ADHD, and to anxiety, and I'll make the perfect test tube baby, right? But just as you're saying, it could very well be the case that, and it's often the case with ADHD, where if you mitigate the unique genetics that encode for ADHD, you're likely also mitigating unique components of that person, like intense creativity, intense dynamism, probably good humor, right? Like just an interesting perspective that arises from that same mechanism that renders them unique.
Thanks, Ian. I appreciate it.
Yeah, thanks for calling in, Daryl. Always great to talk to you. I've known Daryl for quite a while. Hey, Abyss, good to see you, man. 64 gigs of RAM. Jeez, you have a lot of gigs. Oh yeah, and you have an RTX. Yeah, my graphics card. I have like a GTX 1080 i. So it's not bad, but it's not that. No ray tracing for me. It also depends on the gene expression and cell senescence that dictate our physiology. Yeah, for sure. Which is also regulated genetically, right? Senescence. at least in part. Yeah, society calls one thing normal and another not normal, right? Yeah, I agree. Isn't it 30 days or so of repeat behavior causes or breaks those issues? Yeah, I mean, that is sort of like, I've seen that time frame kind of described as like, if you can quit a substance for 30 days. I've also seen 2 weeks, which is almost laughably short for addiction, but for habits, right? Where if you try and impart on yourself a new habit for 2 weeks, you'll get the habit. But for addiction, it's far more complicated than that. We can talk about that after this call. Looks like we're connected. What's your name and where are you calling from?
Michael, and I live in Tampa.
Michael in Tampa. What's on your mind?
I was wondering, I just took a philosophy of mind class this semester, and I was wondering, what do you think the difference is between a philosopher who specializes in philosophy of mind and a neuroscientist? Because it seems kind of interesting, like especially when looking at someone like Daniel Dennett, who's actually postulated some theories that verge more on kind of the neuroscientific.
Mm-hmm. Yeah, man. Well, one of my favorite neuroscientists is technically a neurophilosopher, Patricia Churchland. I think she, when it comes to particularly, you what?
Oh, went over her a lot too. Yeah.
Yeah, right. I'm sure. Yeah, she's very, very famous. And I think, you probably read her interactions with John Searle and I think Thomas Nagel. Right. And the whole qualia debate. From my own perspective, I think she's right and I think they're wrong. But anyways, about qualia. But anyway, so, philosophy. So, like stepping back, right? Science emerged from philosophy. We used to be called natural philosophy. But, it became a distinct discipline once, technology advanced and once a more rigorous sort of pattern of investigation was devised, a scientific method. And so because the brain is so complicated and so unintuitive and so messy, like if you were to make a brain from scratch, you would never make the human brain. It's so messy and just parts of it are ridiculous. But and the reason for that is that they were just good enough, right? They weren't bad enough to prevent us from reproducing anymore, right? So the idea that you're going to be able to intuit various components of human consciousness without, actually doing the wet work of science is, frankly, I think, fairly outdated. And I think that is demonstrated in the fact that a lot of neurophilosophers support, principles that are just not based in science. And frankly, it becomes extremely frustrating when I hear even Daniel Dennett. I mean, I think Daniel Dennett is obviously one of the more articulate neurophilosophers, but they'll make arguments. So, for example, for his argument that that free will does not, or that the mind essentially is not exclusively the non-materialist argument, the non-identity theory argument, that free will and human consciousness can't emerge exclusively from the brain. One of his arguments that I saw him give a talk in college that he made was the afterimage effect, where, right now, if you close your eyes, there will be, the sort of ghost of what you were just looking at, particularly if it was very bright. And it'll stay there for a while. And obviously that's occurring. Nobody would question whether or not that ever even happened. So, it happens. But his argument was that there is no stimuli that are inducing this subjective effect. And so therefore, anywhere in the brain, it's not like the brain is receiving any continuous stimuli from any visual experience. And so therefore, it must not exist within the brain. It must exist somewhere else. But that's just an example of completely not understanding the visual system. That's just not how the visual system works. And, that stimulus is still existing, it's still traveling, it's still being processed within the neurobiology of the visual system. And so it's just a misunderstanding. But he's making a rather important argument using that. It's just one of his points of support. So there's that. There's also the Mary's Room, a thought experiment. I'm sure you guys talked about that. To Or did you guys talk about Mary's room?
Is that kind of the John Searle argument that he postulated? Is it different than the Chinese room, or is it?
It's different than the Chinese room. It's Frank Jackson. Basically,
Okay.
it's the argument that well, John Searle would also make this argument in support of qualia to argue that there are certain components of the human experience that do not exist within the brain. It's just like another example of the anti-identity theory argument. By the way, just so everybody knows, identity theory is not probably what you think it is. It's not like your personal identity, like I'm a white guy from America, right?
Right.
It's that the mind and the brain are identical. That the mind emerges exclusively from the brain. I myself follow the identity.
Yeah, if something like pain emerges, it has to come from the brain, and if it doesn't, then it doesn't have pain. That's what identity theory would postulate, which is why it's not in vogue anymore.
Yeah, well, I don't think it's in vogue among neurophilosophers, but definitely among neuroscientists, yes.
Okay.
At least in my experience, it's not like every neuroscientist I met, I would always ask like, hey, how do you feel about identity theory? But I mean, like,
Well, that's interesting because that's the woman who taught it said that that was one of the silver bullets of philosophy, that everyone stopped believing in it in the 60s. It's interesting that you say that's a pretty substantial difference, which is kind of funny to think.
Yeah, very substantial. I mean, basically, it's like I know you didn't necessarily call to call in to discuss all of this stuff, but basically for me, it's like these disciplines have departed quite a bit. They're Thomas Metzinger, I think, does a fairly good job of sustaining interest in actual neuroscience research rather than just like, but you can't argue against my Chinese room theory, or you can't argue against my so just to give you an example, like Frank Jackson himself no longer believes that the Mary's room thought experiment is explains away identity theory. It's just the more that you learn about neuroscience, about how the brain works, more examples of experiments that have demonstrated a direct relationship between neural activity and changes in human consciousness, the more that these thought experiments are just like, why do we even spend time on this anymore? Like, this is like, it's fun, I guess. There are some I think that are useful. I mean, I do think the Chinese room experiment or thought experiment is like, it's probably one of the more interesting ones. But ultimately, for me, it just seems like so much of neurophilosophy boils down to semantics. Like, how are you defining free will? If you like, Patricia Churchland basically just completely redefines free will, and then therefore says it doesn't exist. But what she really says is like, we shouldn't care about free will. That shouldn't even be a question we're asking. It probably doesn't exist as we, conceive of it. We should think about intentionality and culpability and that kind of stuff. Anyways, so that's been my experience of neurophilosophy. I've always found it very, very interesting, and I've always found it very frustrating. And
I guess, if I could say one more thing before I go.
Please.
One of the things that's always interested me is John Searle has always famously said that he has he understands neuroscience to the level of an introductory freshman student. That's what he knows it as,
Yeah.
It's funny that they always think that he's knowledgeable, but he's not.
And
he really just understands philosophy.
Yeah, and like of course it takes a lot. Like the research that I did, I was using techniques in my PhD research that didn't even exist when I was in college. And so just the speed of development in neuroscience, it's impossible to keep up with all of the new discoveries. But if you're not even up to speed when it comes to like the early 2000s, then like, are you really still going to rely on these thought experiments from like the 70s and the 80s? Like, come on, it's just, anyways, that's my perspective. I would love to see more actual neuroscientists go into neurophilosophy, but it's like many disciplines, very insular. And like, so it's, I suspect it's very difficult to break into that. But anyways, yeah, did you have anything else on your mind? Oh, awesome. Yeah, thanks for calling, man. Good luck on your neurophilosophy class. That was easily, so my roommate in college also went into neuroscience. Neither of us were even interested in science when we got to college. Come back after the same Thanksgiving break, and I was like, hey dude, I think I want to go into neuroscience. And he was like, me too, dude. So we both took neurophilosophy at the same time, and it was so frustrating. First of all, because philosophy students are very good at arguing. Like, they're very, very good at that. That can be frustrating. But it's just it's like trying to, if you want to understand human consciousness, you gotta understand the brain. one of my good friends when I was younger, when it did a PhD in philosophy at, I think, Cambridge. That's like, you know, I don't know, like it's like Saruman's tower of philosophy. Okay, getting another call. Oh, a bit. Sorry. If you don't mind, we'll see if we connect. What's your name and where are you calling from?
Hey, thank you for taking my call.
My pleasure.
My name is Andre. I live in Seattle.
Cool.
Doing software engineering.
Oh, awesome.
Just kind of setting the background. I have a question. So it's a little bit more on the philosophical side, but
Great.
it might apply with neuroscience. Do you think our brain in the future will have the capacity to transcend some of the human traits that we inherently have today, like some of our flaws, like anger, rage, lust, jealousy? And when will we see that?
Yeah, I love it. When will we be able to crawl from the mire of our primate primitive origins? So fundamentally, yes. I mean, I have a screen name. It's Anthropoid, right, on Twitter, and that's sort of like that word is sort of you know, transhumanism, right? It's sort of like kind of a joke that like at some point we'll just be Anthropoid. We won't even be Homo sapien, you know. But anyways, yeah. I mean, I think so, right? Like if you believe, as I do, that human consciousness emerges exclusively from the brain, then therefore, we know the brain obeys the laws of physics, just like any system, and so you should therefore be able to modify essentially any component of consciousness. Now, we're not anywhere close to being able to do that. We're not even close to a completed neuroscience, let alone a completed neurogenetics, right, or behavioral genetics. And so, you know, we're a long way away, but fundamentally, yes. I mean, I think, you know, as a software engineer, like I could imagine a point where at a certain time of your life, you know, you generate an electronic recapitulation of the full anatomy of your brain, where every neuron is, to which other neuron every neuron sends signals, you know, your genetic landscape, your synaptic, you know, your connectome, as we call it, the, you know, specific architecture of all your synaptic connections, make a reproduction of that. And, you know, of course, a bunch of philosophical questions then arise, like, is that reproduction of you, and I mean, obviously you would know better than I would how this would be, you know, would come to pass, would be, you know, come to fruition, but would that thing be conscious? You know, how dramatically different would human consciousness be, human behavior, human psychology, if we're you know, deprived of our corporeal connection to the universe, right? If we don't have to worry about dying, if we don't have to worry about deterioration and time and all of these concerns, you know, would morality completely change? I think one of the best science fiction writers on this is Isaac Asimov. He did, you know, particularly in the I, Robot series, he did a lot of this interesting sort of extrapolation. He was a scientist himself, by the way, but you know, those are interesting considerations, and I could only speculate, you know, very, very loosely. But fundamentally, if you believe that human consciousness arises from the physical, wet stuff of the brain, there's no law of nature, of which I'm familiar, that would suggest that we would be incapable of intervening and modifying and augmenting.
No, I agree with you. I think consciousness in our mind, given that arguably we're one of the most reasonable, or one of the most capable species to reason and plan ahead, I definitely do see a potential for us in the future to
I do too.
take a step further.
It's just it's, you know, we're limited by our technology and our, you know, knowledge of the human brain. But, you know, one of the things that we're pretty good at, we're not great at a lot of things as Homo sapiens, but one of the things we are good at is generating and saving knowledge. And so it's just with a little bit more knowledge, or a lot of bit more knowledge, I don't see why we couldn't.
Awesome. Thank you.
Thanks for calling in, man. If I were starting my graduate career again, I would definitely go into neuroengineering, or maybe software engineering, you know, and join a neuroscience lab. All right. We are now speaking to the abyss. We'll just call you that. We'll leave it there. What's on your mind?
Hey, Ian, how's it going?
Not bad. How about you?
Not bad. So I guess like this is pretty much a general question. Where do you think is like the breakthrough when it comes to neuroscience? Or like which area do you think will have a breakthrough and like in neuroscience in general? Because do you think like it would be more of on the computational side or the neurobiology side? I just wanted to ask like what are your opinions on that?
I mean, I think that we're. I'd be interested in yours too. I think that we're in a period where the sort of the biotechnological advancement is so fast, is so rapid right now. I think it's I wouldn't be the first to say that we're sort of in a golden age of neuroscience, but it's mostly because so many new biological tools are coming out. Optogenetics, hemogenetics. I mean, I'm sure that there's like 12 that I haven't even heard of because I'm not like trying to figure out the next new one that I want to use in my project to stay in graduate school for another 2 years. But I mean, they're just proliferating so rapidly and enabling all these new questions more precise with greater precision, right?
Right.
For me, I would imagine that there will be a couple of events like significantly more effective batteries, these types of things that would enable the integration of devices, in circumstances where, in animal models and so on, circumstances where it was maybe prohibitively difficult to do. I would imagine, like miniaturization, all of these types of things would make it so that the more sort of engineering innovations will sort of become, will proliferate apace, right? In my mind, I think that's, like the efforts like Neuralink is working on, not necessarily them specifically, but some of the scientists that ended up leaving the company, like neural dust, neural lace, these types of innovations that will be a game changer. Oh, and then the neural pixel that I brought up and struggled to remember every time who made it, but it's not Diesroth, it's somebody else, I think at somewhere in California. Anyways, just being able to interface with a wider proportion of the brains of animal models and then ultimately humans, that coupled with dramatic increases in computational capabilities, like being able to manage all those data coming in, tremendous kilobytes like terabytes of data coming in over the course of several minutes, being able to store them, being able to have some kind of machine learning system, I don't even know, to find the signal amidst the noise. I think like that area is sort of ripe for really exploding and fundamentally changing, how we approach questions in neuroscience. So that's why, like, if I were going to go into neuroscience again, start again, that's where I would go.
Okay.
What do you think?
Yeah, because like I'm also being kind of like in a conflict here, because I don't think people would be doing like patch clamp and trying to understand each neuron at a time. And if you consider the computational aspect of neuroscience, it heavily depends upon being able to model the physiology of the neurons. Like you can take your Huxley model or the cable theory model just to apply that how signals transmit along the neuron. But at the same time, when it gets to be like more complex, when you have synapses, it will complicate more things. But at the same time, like what I'm trying to say is that if we can get at least like the nitty-gritty of like how a neuron works, like because as you know, it's a still active area of research. I don't know if it's been like 8 months ago or something, but they actually found out like pyramidal cells are able to compute some kind of logic along the dendritic side, which is like completely unheard of. It's like a Purkinje cell, but they're able to perform some kind of logic operation along the dendrite. But,
I don't think I got that.
So what I'm seeing is that the complexity would simply not end. And if we kind of use the model, the simpler model that we have right now as a basis, I would feel like we would get the computational aspect also flip side, however, you see like the deep mind, for example, having some success, not trying to mimic the brain itself, but by employing more of a statistically sound approach. They use like different probabilistic graphical models to essentially employ some kind of machine learning technique that would look like very simple, like the operation is very simple, but when you have so much back and forth, it would complicate the system, and they're having success in that. Especially, like, I don't know if you heard about the AlphaZero, and he talked about the AlphaFold as well, right? So that could also be an alternative to understand the brain, and they're not exactly copying it, but to understand the perils of how a brain would have functioned if it were tuned in a certain way.
Right.
So I feel like these kind of are like branching out altogether. I don't know if they're going to converge into how to understand how consciousness or like how the brain works in general, but those sort of things that I really constantly keep thinking, trying to identify what the scope is or what the trajectory is when it comes to unifying the computational and the neurobiological aspect of neuroscience.
Yeah, man. Like one of the things that it's such a simple, when we think about like how would human consciousness be, just stipulating that we could reproduce it electronically, how different would that experience be? Because like just something as simple as the fact that like information or signals can travel so much more rapidly electronically than they can in the brain, at least in most cases, almost certainly. And so, we evolved, we were tuned according to a certain time constant, right? A certain a speed at which action potentials propagate, that signals are sent from one neuron to another. And so, would a bunch of systems just totally break just because this structure within the brain is evolved over hundreds of thousands, millions of years to receive signals at a certain rate. And if it just gets hammered by this other brain region that all of a sudden becomes active electronically, does it just sort of, the neurons just kind of are no longer operating, in a way that reproduces biology. I don't know, that is one potential mechanism by which maybe we can't, but I don't know,
But I feel like the neurophysiology itself kind of prevents that from happening, because your spike is determined between a certain thresholds, right?
Oh no, but I mean electronically, right? Right. On the computer.
Okay.
And so those reproduced neurons just don't have a capacity. I mean, of course, the counter argument to that would be we could program that, dude. We can limit the speed of signaling. But still, it would just be kind of an interesting thing to let's crank that up to 10. Let's really, overclock this computer consciousness and see what happens. Yeah, I don't know. It's interesting. Yeah. I mean, I think you're right. Exactly. There's going to be a bunch of sort of independent areas of innovation that all of a sudden oh my gosh, I can use that, and then all of a sudden you just have a far more efficient electrophysiological interface.
Right. And do you think the study of functional groups like nuclei in the brain is kind of exhausted, because you seem to find new regions with more advanced techniques?
Oh yeah.
Like that? Yeah, because that would also kind of eliminate as to how the brain should would function, right? Because if the higher the classification, or if you have aggregates of functional groups, then that can potentially explain how the brain is kind of like a symphony between different or synchronous symphony between different patches of the brain.
Yeah, that is a frustrating thing in neuroanatomy, is that clearly at a certain point we're making arbitrary distinctions, right? And it a lot of these anatomical regions were identified before we even we barely had microscopy, right? It's just literally, hey, this thing looks blue, let's call it the locus coeruleus. It had nothing to do with the functional groups within the LC, but and it happens all the time. There is a group that is proposing that one of the brain regions I studied probably shouldn't be considered an independent structure. Rather, it should be a part of the raphe nuclei. And it's that's fine. They're probably right about that. Whatever. I mean, I don't really care. But there are these sort of arbitrary, it's sort of like, how are you going to sort an Excel spreadsheet? Is it going to be by this, criteria, this criteria? And if you do it by like neurotransmitter similarities, then you're going to have, let's say everything that synthesizes serotonin is called the raphe nuclei. And so there's going to be a bunch, but they're going to be all over the place, right? Not that all over the place, but like, a broader set of regions than is currently incorporated, because that's just a physical, anatomical distinction. But anyways, yeah, I agree. At a certain point, you just sort of get to like, well, let's just characterize every single of the 86 billion neurons, and then they'll all have their own categories, and boom, we can just reproduce that. But anyways, Abyss, awesome to talk to you, man, as always.
Yeah, same here. All right.
Talk to you soon. All right. How long has it been? Oh, man, I got to go. All right, I have a YouTube channel. It's called youtube.com/anthropoid. I talked about Ibogaine, a very powerful hallucinogenic drug that seems to have the propensity for potentially curing addiction, not only to one drug, not only to two drugs, but to many drugs. More than three, actually. So that's youtube.com/anthropoid. And I have a podcast called Wired to Be Weird. Do I watch Joe Rogan? Not as much recently. I used to, yeah. Particularly when he has, people that, he has such a broad range of people that, yeah, for sure. Podcast called Wired to Be Weird. I talk about a bunch of, everything from dreams to drugs to dementia to déjà vu. Those are the 4 Ds that I talk about. But I talk about more than just words that start with D. So Wired to Be Weird. What is a certain neuroscientist? I actually answered that question in the first podcast episode, which is, I think, called What is a Brain? And so anyways, otherwise, I'm going to try and go live again. It looks like they may be shuttering this platform. So if you want to continue chatting with me before they do that, go check out the YouTube channel and youtube.com/anthropoid. So otherwise, I'll see you next week, hopefully, if we're all still here. Same time, same place. And until then, have an awesome week, everybody. See you guys. Oh, wait. All right. So, everybody on YouTube, I passed 200 subscribers, which is super exciting. I had no idea how this would go, but I really appreciate it. And if you haven't subscribed by now, I think it'd be in your best interest to. But I would certainly appreciate it. Otherwise, I hope to see you again next week. See you then.